diversity, where you have the variable regions and your diversity regions and joining regions all coming together. There's a lots of different fragments and sections where this can mix up together and gives this huge amount of potential random diversity that's occurring. Oh, sorry, something. On top of that, you have additional layers of diversity where obviously you have your light and heavy chains, and you have where those two come together and give you another layer of diversity and mixtures randomly. And then on top of that, you have junctional diversity. So the binding and the separation of your VDJ chains together isn't a perfect always combination together, but it's this kind of imperfect process where you can add in additional amino acids or deletions and add another layer of complexity to the sequence layer. And then, of course, the ones that people spend the most time thinking about, especially when we talk about development of vaccines and everything else, is somatic hypermutation. And that's another ability for the antibody, for the B cells when they're seen and exposed to antigen, to actually screen through. And if there's higher levels or better recognition of the antigen, they're much more likely to survive and go on and proliferate. So therefore, there's this huge amount of antibody diversity that's occurring, and that gives us the ability to be much more likely to have a repertoire of antibodies that can recognize and the antigens of interest if you're ever exposed to it. So quiz time. People in the room, because obviously unless someone else can tell me how to get in, look at people who are on with Microsoft Teams. I'm sorry, I don't know how to do this. So I think it's just going to be people in the room. Who wants to guess what is the estimated, just randomly, the naive antibody repertoire? How many potential antibody combinations are there, given that we know that there's a lot of variable regions, diversity regions, joining chain regions in the heavy chain, and then the light chain, there's a whole bunch of different ones, and that's got the variable regions and joining chain regions. And I should have covered this, I didn't, because you don't really need to know the numbers, but like, for example, in the heavy chains, there's over 60 variable regions. The light chains, over 40. Like, those many different combinations randomly thrown together. What's the random approximation, a real true guesstimation of how many antibodies are there potentially able just based on that? Brave guy in the middle.
10 to the 20.
Oh, wow. All right, then you're going to be really disappointed when I share the number with you now. Let's lower the guesstimation, but like a good guess. I wish there was, actually, there very well could be. There very, very well could be, because there are lots of random additional layers of things that could happen. And this is just mathematically what people have been guessing. And it's just kind of the dogma. There are probably other layers of recombination that we probably aren't even delving into, but just, no? Very, very close. There's approximately 10 to the power 12. So that's about 1 trillion potential antibody combinations. Yes, he gets one. This is what happens when you guess. I feel as though we should throw one down to the back, to the guy who was brave enough to guess. All right, so we have this huge amount of antibody potential diversity just through this recombination process. So there's so many opportunities for the antibodies to develop, and there's huge broad diversity. Next guess question. How many B cells are there estimated to be in the human peripheral blood system? Well, yeah, let's reduce our expectations. I maybe I've ramped you guys up a little bit too much. Yes.
I would say like 2 million.
Closer. You want to go a little bit higher?
I'll give this one to this on my home.
All right, we'll go here. So this is the guesstimate. There are about 5 billion. So you have trillion recombination, based on recombination events could occur. And in the human peripheral blood B cells, again, this is just a random estimation. probably a huge amount of diversity as well in the spread, but people estimate that there's about 5 billion B cells. So if you're thinking about that mathematically, that's a tiny percent of the potential antibody repertoire that's actually available. So there's a lot of people who spend a lot of time thinking about this and going into like understanding, wait a second, if there's 10 to the power of 12 potential combinations that are occurring. Of course, some of them are disappearing, and well, there's a whole bunch of other stuff that people aren't even, I'm not even going to cover about tolerance and XYZ and deletions. But only about 5 to 10 power 9 or 5 billion cells, then how much chance is there if the person next to you, do you think there is in the similarity in your B cell repertoire? Literally the person or the closest to you. Are you highly similar or are you very, very diverse? You're pretty diverse, right? And that's just the amazement of the B cell, of the antibody system. And that you still, if you think about it though, vaccines still work. And what's driving that? What's going on here? And there's some really interesting studies that delve into this and think about it. But one of the ones I just want to very quickly cover, and I think it's just a fascinating concept, is this group that was led by Burton's group. What they did is they took 10 individuals, just random 10 individuals, leukapheresis them, leukapheresis, and sequenced as much as possible in depth the entire antibody repertoire. So I think each of them, they got approximately 1 billion sequences each, rounded up. And then they looked at those 10 people and said, how much similarity was there in B cell, in antibody clonality was there? And when you actually start thinking about it then, you actually realize that for all 10 individuals, 0.022% of them had shared clonality. But if you think about it some more, that's actually quite a surprisingly common amount, given that there's this huge potential antibody repertoire and everyone has a limited number of B cells, of which everyone is actually sharing lots of similar clonality amounts. So therefore, maybe vaccines are working really well. And it also demonstrates that your general peripheral environmental and development is actually harboring and developing very similarities, considering the huge repertoire of antibody recognition and sequences that are occurring. And I think just one really interesting way that they demonstrated this is looking just specifically at the CDRH3's regions and positions. If you look at them, there were 3 shared nucleotide mutations amongst all 10 people versus this number of sequences where there were 2, where there are 3 amino acid mutations or nucleotide mutations amongst the 3 people in comparison to this many of sequence similarity or differences in the 2 people. So there's huge spread, but we are still able to actually share similarities in the antibody responses. So I just want you to realize how amazing antibodies are and how can we harness this and how do we develop antibodies and make sure that they are targeting the right things so that they are hopefully are protective and not pathogenic. So as you may recall, as mentioned very quickly, you have that Fab region, and that's a region that recognizes the antigen that binds to the target. And in the case of, for example, vaccines, we spend a lot of time thinking about neutralization. So we think about the ability for the antibody to bind to those really rare and unique sections of the pathogen that will, say, neutralize the toxin, or will block the ability of the virus from entering or preventing it from infecting or causing damage. Now switching to the completely other side, and this is the part that I spend my time spending the most time thinking about, which I think is most interesting and actually has maybe not quite as much diversity as the Fab region, but still a huge amount of diversity, is that FC region, that crystallizable region, or the constant domain. And this is a region where it actually mediates a lot of different functions, ability to kill, ability to activate the surrounding innate immune cells. And the way that they do this is uniquely that these antibodies don't have to recognize these key epitopes or these key targets that specifically block the virus or the pathogen or the toxin from causing damage. Instead, they can recognize any area of the pathogen or any target. And this is where it's particularly beneficial when we talk about viruses where there's high levels of mutation, because it can actually recognize maybe the more constant regions or the similar or conserved regions of viruses, and may be beneficial in that part because they can share that level of similarity and able to recognize, for example, when we talk about COVID, different variants of concern where it's not specifically targeting the highly mutable regions which are often trying to escape. And the unique thing about these antibodies here is they can bind and they create immune complexes. So that means multiple antibodies binding to the targets, and then when their immune complexes are forming, they can therefore activate surrounding innate immune cells through engagement of Fc receptors. So Fc receptors are these surface receptors on innate immune cells, and then you're thinking about things like dendritic cells, monocytes, phagocytes, NK cells, and dependent on the innate immune cell there, that immune complex formation, engagement of multiple Fc receptors causes activation and sometimes inhibition, but mainly we're thinking about activation, and then results in a range of different functions such as secretion of chemokines, killing, engulfment, phagocytosis. So what defines the ability of these antibodies to drive these different functions? And that's what our group really is interested in looking into. And that's the differences in the Fc structure. So in the humans, we have this gene loci where there are different, you've probably heard of class switching that occurs. And there are different isotypes. So you're very familiar with IgM, D, G, A, and E. So the most common one that you're probably very familiar with is IgG, that middle guy. It's the most abundant in the blood. It's the most prevalent, it's present, and it's the most functional, and I'll spend a lot more time talking to you about it. But you're probably very familiar with IgM. That's probably the most early type of antibody, and it is highly cross-reactive, and it's also given a special avidity due to its pentameric structure. So when you think about functions, IgM is great, especially with activating complement. And that's because of its pentameric structure. It's really great at then activating complement, and the complement forms these hexamer structures and activates them, and the classical pathway downstream there. You also have IgE, and IgE you probably are familiar with when you think about allergies, and they're traditionally known, for example, activating mast cells. They're the reason why I have really bad hay fever, for example. High levels of allergens, antibodies to IgE target them. They're really also really important when you talk about parasites and clearance of parasites and that manner. And then you have IgA, and IgA, you should probably recall, is an antibody that's most present in the mucosal regions. It can come in different forms. You have your IgA monomers, and then you have your dimers. And the dimers are most prevalent in the mucosal surfaces, and then your monomeric versions are most prevalent in your blood. And then when we delve into understanding IgG and IgA even further, there's a whole bunch of different subclasses furthermore. So IgG and IgA have a range, or at least humans, have a range of different subclasses. And this is really interesting because each of these seem to have evolved in humans to do different things. So your IgG1 and IgG3 are actually most people think of as the most functional when we talk about the activation of Fc functions. These are really great at binding to Fc receptors, activating those different functions that I mentioned earlier, great at binding to complement. In particular, IgG3 is really unique, and I spend a lot of time thinking about it. I'll delve into a little bit more about it later on. But you can see here what we've drawn is this really extended hinge region. And this means this hinge region gives this antibody a lot more flexibility. So you can imagine it's kind of like, you know how you go to, or you drive by those old cars or second car salesman, and you can always see those flapping guys or those windy, you know what, those wind tunnel kind of things. It's kind of like that. It gives it an extra level of flexibility, and therefore, unlike other antibodies, it can actually arrange and flex in such a ways and actually reach in to target different epitopes that a traditional IgG1 can't target. And monoclonal antibodies, where they've been made into IgG1s versus IgG3s, have actually demonstrated, because of the extra flexibility, they actually sometimes have better avidity or better ability to neutralize, just purely because of that greater stretch and ability to bind and reach targets. IgG1 and IgG3 are also the most prevalent when we think about viral protection. And the reason being here is that because IgG1 and IgG3 generally bind to proteins, you see that highly expressed in recognition to protein antigens. On the other hand, when you think about IgG2 is often seen and developed to bacterial pathogens. And the reason being is that IgG2 traditionally seems to be expressed and binds better to carbohydrate structures. So you may see that IgG2 actually is really beneficial for protection against bacterial pathogens. And then when we look into the ability of the IgG2, you can see that depending on the Fc receptor, it can bind, and I haven't delved into the different Fc receptors yet, it can bind with variable affinities and activate a range of different Fc receptors accordingly. And then IgG4 is really fascinating as well. So IgG4 here is, I mentioned it, I'll go into, because I think I've got another slide about this, is unique because it's often induced in allergies. So people who have studied beekeepers, for example, so these are individuals that have been exposed to bee stings repeatedly over and over and over again, and especially people who originally had allergies to bee stings and are beekeepers, what they've seen is that they have actually tolerized themselves to the bees, to the point that what they do is they overexpress IgG4 levels to the bee, whatever that bee sting toxin actually is, and you have these huge amounts of IgG4 levels that are observed specifically in beekeepers because they've developed this tolerization to reduce the ability of the other antibodies to actually have that activation capacity. And this is because IgG4 is a very, very unique protein, which I might, because I got ahead of myself because I'm so excited about IgG4, delve into. What's really interesting about IgG4 is that it, depending on the Fc receptor, it can bind okay or pretty poorly in comparison. But the thing that I find the most fascinating about IgG4 is that it's bispecific. So unlike all the other antibodies, and I'm going to delve into here, where you can see, we probably should have drawn this better, have this hinge region where you have disulfide bonds that link them and hold them together, those 4 chains together. IgG4 is the only really interesting antibody that doesn't have the disulfide bonds. So in the actual body, people have described, I actually have to admit there's only one study that have described this really, really well, is that they can actually mix together. And you have, therefore, in the general blood, people have described, well, one study has described really well, the characterization that these antibodies potentially can actually recognize 2 different antigens at the same time. So there's a natural bispecific capacity. And because of that, there is a potential here that they're actually contributing to that tolerization or reduced anti-inflammatory capacity, because there's a less ability to cross-link. Because, for example, it's great if it's to the same target antigen. So if they're 2 to the same type of virus, then that's great, because you can imagine that the linkage can occur. But alternatively, if it's bispecific and it's binding to very different antigens, one which is a flu and then the other one which is a bacteria, then the opportunities for those 2 to occur may be less likely, and there's less chance of them forming immune complexes. And in addition, because there's less ability and weaker ability to bind and engage Fc receptors, it's been described to be potentially inhibitory and reduces Fc effector functions. On top of that, you have your IgA1 and IgA2. IgA1 is a very interesting structure. I do want to mention IgA1's interesting evolutionarily in that only humans have it, like humans and chimpanzees. If you look at the mice, for those people who do antibody mice, you probably are thinking, wait a second, are there subclasses in IgA? The answer is no. And the reason being is that for some strange reasons, humans have maintained two subclasses of IgA, whereas other mammals wisely, or maybe not so wisely, have evolved to get rid of it, because IgA is actually really sensitive to a lot of enzymes. And what IgA2 is actually quite resistant to a lot of enzymes. You might probably have heard of enzymes that are like ENDOS and etc. that you actually maybe even using biochemistry actually are very sensitive to it, whereas IgA1 is very sensitive versus IgA2. So the IgAs that are most similar to, when you're looking at, for example, mice, is most similar to the IgA2, whereas the IgA1 is very, very different. And this is all due to these differences in the hinge and the Fc structures. And you can see that IgA1 has actually a different spread. So a lot of people argue that IgA1, because of the greater spread, similar to the IgG3, may actually give it greater ability to flex and bind and a greater avidity, but the downside, as mentioned, is it's more sensitive to enzyme degradation. And then there's another layer of diversity that we think about, and that is genetic diversity. So you're all probably very familiar with MHC diversity, and you're all probably very familiar with like Cure diversity when you're talking about NK cells. Antibodies have a huge amount of diversity as well. So there's a large number of different allotypes that have been described, and these are inherited ethnically, and I'm not going to go and delve into them. But what's really interesting is that, in particular, IgG3 is the most diverse of them all. There are over 15 allotypes that have been described. Mentioned, they seem to be inherited ethnically or geographically. And depending on which IgG3 genetic allotype there is, there's some that increase the half-lives. So IgG3 actually naturally has a 7-day turnaround, whereas IgG1, for example, has a 21-day turnaround. So that's a big difference. But if you are one of the people who has that rare gene, your IgG3 hangs around for 3 weeks. So that could potentially, and these differences have been associated with different diseases as well and autoimmune responses. It also affects the way that they bind to FcRn and are transferred across the placenta. We know that dependent on the different allotypes, they actually bind to Fc receptors and complement differently because of those amino acid changes. Especially complement is very sensitive to different allotype changes. And then when we also delve into this in further, dependent on the allotypes, people have described that specific allotypes, because they're inherited genetically and ethnically, have been associated with better protection against different really long-lasting or those diseases that have been followed and inherited over time. Malaria is one in particular. So if you look into unique areas in Africa or regions where there's huge amounts of malaria, there's been associations with particular maintenance of these rare allotypes in these regions which are really high endemic with these pathogens, for example, malaria. And the reason why this is interesting is because it's from this information of IgG3 that some of the information about the development of antibody and the structure of the Fc regions to improve therapeutics has been experimented with, because these high amount of diversity gives you clues to amino acid changes that improve the better binding of Fc receptor responses, skipping through the IgG4. And then another layer, which I find very interesting, is also that not just the genetic structure changes, but there are also post-translational modifications that changes. So you probably really remember back in your immunology classes that it's not exactly B cells that are producing antibodies, it's these antibody-secreting cells. So these antibody-secreting cells are the ones with huge amounts of cytoplasms and huge amounts of ERs and the reticulums. And this is necessary because they're producing huge amounts of protein, but these proteins also have huge amounts of glycans. And every IgG in the body, regardless of 1, 2, 3, and 4, in the Fc region has this unique asparagine position where you have this N glycan added to it. And what's really fascinating about this is that dependent on this change or the presence or the size of this little sugar moiety that's added to this one region, it completely changes the structure of the Fc region. So you can imagine that here, the reason why I like this shape here is that you can see that if you were to make this a little bit smaller, you can imagine that maybe the Fc region would be a little bit slimmer. And studies have actually shown, actually crystallized this, where you can actually have demonstrated that the presence or absence of specific glycan structures, the most well described is this fucose structure, is that if you cut off that fucose, this little red triangle, it actually means that the Fc region doesn't induce, is a different shape, and it actually can bind into Fc regions with much better ease. So there's a reduced steric hindrance that seems to be occurring very basically, just due to the fact that this little tiny bit of the sugar structure is being removed. Monoclonal antibody therapeutics have really delved into this, explored this, and have shown that if you actually cleave that fucose structure, it improves ADCC binding, so antibody-dependent cellular cytotoxicity, about 50 times more with or without the presence of this little tiny sugar. And this is something that the monoclonal antibody therapeutic area is actually delved into. Studies have shown that, for example, for rituximab, you might remember this as a cancer monoclonal antibody for B-cell lymphomas. It binds and recognizes CD20. What actually happens is that if you use and have an antibody with a normal Fc region and normal levels of fucose versus those that actually cleave off and reduce and significantly reduce fucose, the efficacy and the therapeutic benefit of these monoclonal antibodies is so much better in the clearance of these B-cell lymphomas. And then on top of this, you have other layers of regulation. So these little grey ones is galactose. And if you have a shorter one, so removal of these galactoses or presence, these are much more associated with autoimmune diseases, and it's also being associated with disease progression and inflammation. So people who have autoimmune diseases, people who have chronic infections, they generally have much shorter galactoses and shorter structures. On the other hand, you have this other blue thing, and this is a sialic acid. And sialic acid is a fascinating one because it's been associated with anti-inflammatory properties. So the opposite of these, the longer these kind of sugars are, and the glycans are more branched they are, they're more anti-inflammatory they are. And we know this because our bodies naturally regulate this. And in women in particular who become pregnant actually upregulate and change their total antibody glycan profiles. So as mentioned, autoimmune people have these shorter glycan structures. When women become pregnant, to kind of protect themselves and reduce the inflammatory processes that may occur in the bodies, what they actually do is they significantly upregulate the layers of sialic acid. And it's this protection measure such that it means that they don't have this excessive inflammation and protects the fetus and excessive inflammation that occurs. And what's really fascinating is that when people delve into look at, say, women with autoimmune diseases, when they become pregnant, they go into remission. What's happening is that the women are all naturally regulating their bodies and upregulating sialic acids in their antibody structures such that they are tampering down and reducing that inflammatory process. So just the presence of these sialic and structures, it seems to do quite a lot in the general antibody systems and what's been associated with remission in autoimmune diseases. But on top of that, this has been used as therapeutics. So this is, some people suspect is contributing to, say, IVIG therapy. So you probably have heard of intravenous IgG that's been transfused into autoimmune individuals. So these individuals have much higher levels of, the IVIG has much higher levels of sialic acid, and that helps to reduce, some people think the mechanisms, or one of the mechanisms, is that tampering down of the inflammation that's occurring. Oh, sorry, did I do that twice? So in general, you can see that there's multiple layers of regulation, just thinking about the FC region, not to even mention the Fab region, that just blows your mind away, that can happen to the antibody. You have your antibody structures with the FE region, where you have this huge amount of diversity and recognition of targets, where you can recognize different epitopes, and also somatic mutation that occurs with higher affinity. You also have the ability to recognize breadth, or as mentioned, like maybe the targeting more conserved regions versus more specific regions. On top of that, as mentioned, you have your FC region, where obviously the structure layers are occurring. You have your isotypes, but then a lot of the isotypes are different subclasses, as described beforehand. You have the different glycans that are also modulating what's actually happening and how they're binding to FC receptors, and your different glycoforms. In humans, there are over 30 different glycoforms. So I was just giving you like a tiny little summary of some of the things that can happen, but you can imagine there's lots more that can occur for the glycans. And then, as mentioned, the genetic structures that can occur. And then on top of that, you have your FC receptors. And I haven't even gone into this, but in humans, you have a huge amount of FC receptor diversity. So dependent on what isotype, subclass, and that profile that you have, it will actually contribute to which FC receptors it can engage. So you have your FC receptor 1, 2s, and you also have your receptors that bind to, say, IgA, or IgE, which are completely different FC receptors. And they are all expressed in different layers on your range of different effector cells. So the reason why I suspect that people know a lot about, or you may have heard about ADCC, or antibody-dependent cellular cytotoxicity, is because people spend a lot of time looking at NK cells, because I have to say, they're pretty easy to understand in the FC receptor field, because NK cells generally only express one FC receptor, FC gamma receptor 3A. I do want to give you a little side note that about 20% of the human population actually express 2 different types of FC receptors in the NK cells, this thing called FC receptor 2C, but most of them are expressing this FC receptor 3A. On the other hand, you have these other cells, neutrophils, which are really complex, and they can actually express pretty much every one of these FC receptors in different layers. So how do you determine what the antibody and this is doing if you have dozens of different FC receptors on the cells? And that's why understanding how FC receptor functions affects, say, a neutrophil is so much more complex than understanding what an NK cell is doing, because it's a single type of FC receptor. But altogether, this contributes to dictating what type of function or immune response is being delved into by the antibody. And just very quickly, because I'm aware that we're running out of time, is what our group is really interested in is delving into that complexity and understanding how do the different parts of the structure and that kind of those little tiny changes of the FC region or the Fab region contribute to dictation and modulation of antibody functions. So what we do in our group is we run assays and we profile the antibody glycans, delving into those different structures and how they better or worse bind FC receptors. You also have the different subclasses binding to the different, how well do they bind to different FC receptors, how well do they bind to different epitopes of interest, and then a range of different actual cell-based assays, understanding, are they activating complement system? Are they activating different FC effector functions, like is it ADCC response, a killing response, or alternatively is it a cytokine response, or is it a phagocytosis response? And then we combine all that data together and create these humoral fingerprints of understanding what's actually happening and summarize to understand what's driving these responses. So very quickly, just an example of the studies that we have been doing is we've been looking, for example, this is an old study, HIV vaccines. So this vaccine was done in Thailand. It's called RV144. You don't need to remember it. Just remember it as a red vaccine. So the red vaccine was moderately protective against HIV. This is a human vaccine. You're never going to roll this vaccine out. It's only 30% efficacy. But it's enough for us people to want to delve into and understand, wait a second, what about the antibodies potentially are providing protection? And can we ramp up that type of antibody? in the future to improve vaccines and have a higher amount of that antibody so that we can improve the protection instead of 30% to maybe 50% or something higher. And then what we did is we compared it to its predecessor. So this vaccine is the blue vaccine, and the blue vaccine actually consisted of the exact same protein component that was used in this protective vaccine. So this blue vaccine is very interesting, and I give a nod to people who were volunteered for this trial, because this vaccine included multiple doses of protein vaccines over 30 months. You can see here 1, 2, 3, 4, 5, 6, 7 doses of vaccination. But we were delving into, this was a non-protective vaccine. We wanted to understand what are the differences in antibody responses between those two groups. We ran system serology, ran a whole bunch, understanding the different layers of the structure. What does it target? What are the FC regions' protein structures? What are the how well does it bind avidity-wise? What cells does it activate? We get these huge profiles together, and then what we do is we run computational analysis and then delve into identifying what are the features or what are the unique signatures that are really present in the red vaccine, which is protective, versus the non-protective vaccine, which is blue. And we kind of screen through and understand what's better and elevated in the protective vaccine. And as you can see here, red groups together, protective, blue, non-protective groups together. And then what we also delve into is understanding what are the unique signatures, what's really present and distinguishing the two different groups. And we see here is this IgG3 is the most and the strongest antibody distinguisher of this protective vaccine. So what is IgG3 potentially doing? As mentioned, as a reminder, IgG1 and IgG3 bind to Fc receptors really, really well in comparison to the other subclasses. And we can see here, when we look at how they correlate, this is the IgG1 and then IgG3 across the x-axis versus the y-axis. There's these different Fc effector functions. Red means it's highly correlated. So the red vaccine, the RV104, IgG1 and IgG3 really strongly correlate with Fc effector functions. And therefore, the vaccine seems to be driving this elevation of Fc effector functions that may be protective in this vaccine, and specifically this huge amount of IgG3. On the other hand, the non-protective vaccine, we see some levels of IgG responses, but it lacks this polyfunctional ability to mediate a lot of Fc effector functions. And what's really interesting on top of that is when we deplete out the IgG3, we can see there's a significant drop in the ability of the vaccinees to mediate these responses. So IgG3 very clearly is key for driving and contributing to Fc effector functions. And then other studies have actually demonstrated that IgG3 really correlates really well with actually protection from HIV in these case control studies. So on the other hand, as mentioned, IgG4 is an area, as a subclass, that really intrigues me. This is the one that you might remember as a bispecific. It has a potential to recognize two different epitopes, and then on top of that, it binds pretty poorly to Fc receptors. And what we saw is that when we pulled out the IgG4, especially in individuals with the blue vaccine, where we saw there's huge amounts of IgG4 in comparison, this actually resulted in an improvement in Fc receptor responses. So the IgG4 is actually detrimental or potentially inhibitory or reducing IgG4, and therefore it may be tolerizing to the vaccines. Now I'm just going to realize that I'm just overestimated what I could cover in a lecture, so I'm just going to skip straight ahead. I'm sorry. What I'm just going to very quickly cover is we have done a whole bunch of work in the COVID field and understanding, and everyone, as you know, has been vaccinated with COVID-19 vaccines. I'm pretty sure nearly everyone in this room has probably gotten at least 2 vaccines, maybe the 3rd vaccine. But one really interesting thing that we've seen, I'm sorry. Yeah. I overestimated what I could cover. I'm sorry. Is what we see is we see very different profiles in antibody responses dependent on your antigen exposure history. So what do we mean by antigen exposure history? So this means that dependent on whether you have been infected with COVID, because as we know, there's very few novits left in the world, or alternatively, what type of vaccine you're given can actually completely change your antibody profiles, whether it's the type of isotype or even subclass, and the doses and all those types of formulations seem to be driving different antibody responses. And what we've actually delved into is really understanding what does the different antigen exposure history is doing to mediate improved or different Fc receptor responses. And one study here, what we're doing here is we're looking at individuals that have previously on the left been infected and then got vaccinated. On the other hand, those people who just got vaccinated. And you're seeing very different antibody structures. I'm not going to delve into it in huge detail. Just the note that, wait a second, there's better Fc receptor binding and better Fc effector functions in those people who surprisingly have got infected then vaccinated. But I just want to come back to this IgG3 and IgG4 story. And the reason why I think this is fascinating is that individuals that get 3 vaccinations of an mRNA-based vaccine suddenly have this huge elevation of IgG3, but this huge elevation of IgG4 responses as well. And the IgG4 responses in particular are extremely high. So as a reminder, I was mentioning IgG4 is a really interesting subclass. It's this bispecific potential subclass that binds to Fc receptors a little bit differently than the other subclasses. And multiple vaccinations is inducing much higher levels of IgG4. As a reminder, when we deplete out the IgG4 is correlating with worse Fc receptor responses, and you have higher, and when you pull out the IgG4 and deplete it out, it actually improves your Fc receptor responses. So what we've actually seen is that individuals that have been vaccinated multiple times with Pfizer vaccination actually have high levels of IgG4, which actually inversely correlate with Fc effector functions. So therefore, multiple vaccinations are driving repeater antigen exposure, is causing more class switching, and therefore it's driving downstream activation to this IgG4 subclass that binds or poorly binds to Fc receptors and actually results in a dampening, and some people argue potentially maybe tolerizing your body to SARS-CoV-2. And therefore inversely correlates with the Fc receptor responses. I'm just going to skip through here. You can see I'm really overestimated what I could cover. I'm sorry, guys. I had so much stuff I wanted to cover, but I got excited about other stuff. So really what I just want to cover here is that obviously there's a whole bunch of different layers in which antibodies can be modulated. And this can be modulated dependent on your antigen exposure. Maybe what I will do is, for those people who are interested, I didn't put it in here. Just for a heads up, for those people who have actually got AstraZeneca and then Pfizer vaccination, you guys don't have huge amounts of IgG4. You guys have normal levels of IgG4 and you don't have this Fc receptor binding. I'm sorry, you can see I've skipped something here. But you can see that there's huge amounts of depending on what type of antigen exposure and different vaccine modalities, infection versus how the formulations or the types of vaccination, you can completely skew your antibody profiles. You can change them to your isotypes, you can change your subclasses, you can change the effector functions to boost different types of antibody structures accordingly. And I think I'll end off there and thank a lot of people who have contributed to the work, and I'm very happy to answer any questions about antibodies in general.
Thank you so much for the wonderful seminar that helps us dive into the world of antibodies. So yeah, now it's question time. For those who join online, you can also just type your question in the chat. We will also actively monitor the chat section of Microsoft Teams. So any question?
Hey, that was great. Thank you. So is there actually any advantage to having the focus on the Fc region of the antibodies? And why do you think we evolved it?
So it's great because it reduces activation. So you can imagine if, and people still, and this is a really hot topic right now, is how do we modulate post-translational modifications? How do we regulate your B cells or your antibody secreting cells to have different sugar structures. And if it has more of those fucose, wouldn't that actually be a good thing if you had autoimmune disease, for example? Because there might be less activation in that sense. So it may be beneficial, and I think it's really dependent on the situation. Do you want excessive activation or do you want less activation? So dengue disease is a really interesting disease where people have shown that if you have really excessive amounts of antibodies that bind to a lot of S receptors, it actually contributes to severe disease. And people have shown that those individuals with dengue disease actually lack the fucose, and that actually contributes to overactivation. So we want to make sure that doesn't happen. It's a really interesting balancing act.
One more question, if people don't mind. So the increase in the IgG4 levels after 2 doses of vaccination and the booster and compared to natural infection and then vaccination, has anyone gone back to challenge those people with COVID just to see what the clinical response is like?
Yeah, I wish we could do that. I can imagine the ethics would be painful to do that. Human challenge models. I do know that there are actually human challenge models that have been done, but they, to my knowledge, the ones that have been done in very unique settings where there have been very small, highly monitored settings where, for example, I know Imperial College has done human challenge models, and they were all naive or at the time was naive. I don't know, but so when you say challenge them, alternatively, you could just think about monitoring them and seeing, do they have infection later on? Is that correct? So rephrasing a question, do you think that those people who have been vaccinated and then got infected again have different, can you paraphrase what you mean?
Like, say, a quicker clearance of the virus or, any sort of clinical correlate?
No, I don't know. I don't think there's any study that has delved into that in great detail yet. This IgG4 thing is something that people have just observed or published pretty much at the start of this year. And I think it's an area that we're really interested in delving into because worldwide, there are now literally millions of people walking around with huge amounts of IgG4. And then on the other side, I actually wonder, is IgG4 beneficial? Because we know that in severe infection with COVID, and I might be delving a little bit too much into virology and how COVID works, but you probably might remember those people who have been following the COVID stories. Severe infection has been associated with this cytokine storm and this over-inflammation in the individuals that progress and pass away or elderly individuals. So what if IgG4 is actually beneficial because it's tolerizing your body and reducing the inflammation or reducing the chance of you actually developing severe disease? That's just one random hypothesis. There's multiple conflicting stories about what IgG4 could be and could not be doing, but it's one thing, and it's really interesting. And delving into understanding IgG4, I think, is really interesting area and hot area right now.
Thank you.
So about the IgG4 stuff as well, for basic understanding, I also wanted to ask, with the class switching, sort of how do you, what signals are there to actually decide what class you switch to? And also the other thing was, is IgG4, it probably isn't really well known at the moment, but is it being switched to because it's beneficial, or is it just being switched to because you're getting repeated exposure to an antigen? And as you get more and more exposure, maybe any antigen would skew towards IgG4.
Great, two great questions. All right, I'll start off with the last one because I remember it the best, and I might ask you to repeat the first one. So repeated antigen exposure potentially could be it. But then it got me thinking, going, why don't we have huge amounts of IgG4 to influenza? Does anyone study influenza here? Has anyone delved into looking at subclasses in influenza? We know that people get infected with influenza over and over and over again in their lifetimes. Is there huge amounts of IgG4? Yes, there's higher levels, but is there levels that we see for the vaccines? No. So I definitely think there's a level of repeated antigen exposure involved, and that obviously that's been described really well for bees, like in the beekeeper stories and in tolerization. But there seems to be some other layer of regulation that's occurring, and it may be that maybe infection is a very, very different system that's occurring that's driving obviously a lot of other things that are happening in the immune response that means that you don't get that versus being exposed to one particular repeated antigen without certain stimuli, maybe driving something else. And it's an area that I'm really fascinated about and really, I think people are really thinking about right now. The first question was?
It was just sort of a more basic thing of
class switching. Yes,
yeah,
that's
how
right.
class switching, how do you decide?
Yeah, so class switching is, again, really complex. There's a reason why I skipped it, because I was like, I could spend like 20 slides on this. So class switching, there's T-cell dependent and T-cell independent class switching. We know that T-cell dependent ones are really important. So people think about TFH cells, and we think about the contribution of T-cells in regulating that. But there's also this new area of research which has demonstrated that innate immune cells are really drive it. So it's very seen to be cytokine based, and T-cell independent class switching has occurred, where you see, well, actually, maybe let's take it back. Gamma delta T cells are T cells. So you can have gamma delta T cells and NK cells contributing to class switching, but you can also have demonstrated, at least in mice, you see neutrophils driving class switching as well. So it seems to be multiple different signals that contribute. Very large driven by cytokines. So dependent on different cytokines, in humans it's a more complex process because people haven't been able to delve in as much detail and understanding the regulation because we can't do in vivo experiments. But in vitro, you know that different cytokines plus activation of different receptors can drive different signals. For example, IgG4 has been really well described to drive IgE. IL-10 has been known to drive different combinations of subclasses. So there's multiple layers in which it can be regulated.
Sorry, that was a great talk. Just again with IgG4. You spoke about like IgG4 having a potential role in modulating the humoral response. Or has its role been studied in, say, autoimmune diseases versus active and then refractory infections? And might that help elucidate what it's doing in the body?
Yeah, definitely. So there's actually IgG4-driven autoimmune diseases as well. So that's also this detrimental element of having huge amounts of IgG4 for sure. And I think the other thing to note is IgG4 is really interesting and trying to understand what's actually happening in vivo with antibodies in humans is really quite complex, given that antibody subclasses seem to be very different in different mammalians. So there is no perfect identical IgG4 in mice or IgG3. Actually, there's no direct similarity of the different subclasses in humans as there are in mice, because the perfect way would be then to prove what IgG4 is doing in mice and then looking at that. But mice, I don't know if people have studied mice or understand subclasses in mice. In the IgG structure, you got IgG1, you got 2A, 2B, and IgG3. IgG1, if you really want to like squint, is a little bit like IgG4, but it also binds Fc receptors as well. IgG2, A and 2B are most similar to your IgG1-ish and 3s. And it's really quite complex. And IgG3 in mice is just this weird subclass, and we don't even know what it's doing. But ideally, I would love to be able to create a model in mice and say, IgG4 is this subclass is most similar. If anyone can make a humanized mice that can do this, you guys would be amazing. And delve into understanding what is IgG4 in a vivo mouse model. That would be brilliant.
Thank you.
Could I actually ask a question? Wait. Sorry. Yeah, I just have a general question about the classification of those constant region subclass. I'm just wondering, is this kind of different kind of antibody subclass, like IgG1, IgG2, is actually naturally stochastic? So like, for example, there could be something existing between IgG3 and IgG4, maybe let's call it IgG 3.5, because we can see there are lots of kind of subclassification already, such as the allotypes, post-translational modification. I'm just wondering if we just keep this kind of like classifying them into this kind of categorical classification, would there be actually infinite amount of different antibody subclass, this kind of subtypes?
So maybe there's definitely a huge amount of genetic diversity of IgG3, for example, but a lot of these diversity that's occurring, we're talking about some of them are as simple as one amino acid change. Some of them are a string, 10 or 15. Some of them are very, very, very large. And especially in changes in the hinge region, you can be talking about like dozens, but we're not talking about sequence homologies where you have huge amounts of changes. So I'm not sure if there'll ever be a 3.5 or 3 point, etc. But on the other hand, there is still a huge amount of diversity. And I think when we think about this here, as mentioned, there's about 15 that have been described. There have been studies, more recent studies, because these studies have been done mainly in Caucasians. Studies that have been done very recently, like the last decade, profiling Brazilians, like rare populations of Brazilians, they've found over 30 different other alleles just in Brazilian individuals. And you can imagine people who, some people, even in Lehigh, are probably aware of this, that there are people who've been looking right now in allotypic differentiations in more rare error regions in PNG. And you're seeing a huge amount of diversity there as well. So it just may be just representing the fact that we just haven't looked and really sampled the population to see these differences. But I'm not sure if we'll have a 3.5 to our knowledge yet.
Thank you for that answer. Thank you.
Thanks for that great talk covering the large complexity involved. So I'm very interested in the post-translational modification aspect, and you said that this is being attempted to be targeted therapeutically. How would it be targeted? Would, say, like there's a protective function of having glycosylation at a particular site, say, or maybe the opposite. Would you just have an antibody with a mutation that would prevent it from being modified at that particular residue? How are people starting to think about addressing these PTMs therapeutically?
Yeah, that's a really exciting area of research. So what people have seen in vaccination is, if you look at, and this is in human studies as well as animal studies, if you vaccinate the individual and look at the antigen which has been vaccinated with, and then put in different adjuvants that activate, say, a toll-like receptors or different cytokines and stuff like that, what you can see in parallel, you can monitor other, what we call other targets that aren't present in the vaccine, and look at their glycans, and then you can look at what are the glycans and how they're modulated from the actual antigen of the vaccine. And we can see that the vaccine itself seems to be modulating antigen-specific glycan changes, whereas, for example, there's been these human studies where we looked at vaccination of HIV vaccine, and in parallel just monitoring influenza antibody responses. The influenza antibodies don't actually change much by the vaccine. On the other hand, the vaccine, where you play around with the different adjuvants that have been added to it, have different FC glycans on them. So very clearly that means that it seems to potential to be antigen-specific modulation through the adjuvant of interest, whether it's activation of toll-like receptor or a certain cytokine added in combination. Sorry, I take this back. It's not humans. This is a macaque study. But you can actually see the same thing where you, for example, looking at the earlier vaccine where I was talking about vaccine 3 and alpha-1 where they're the same antigens being exposed, looking at the glycans there, you can actually see different glycans present in those two different vaccines. How are we thinking about it in the bigger picture besides vaccination? I suspect epigenetics will be a very interesting area. Understanding how do we, and I think a lot of people are interested in this, like how do you modulate the epigenetic changes that could be contributing to it beyond the adjuvanting of that? One area of potential interest.
Amy, did I hear correctly that IgG4 doesn't have a disulfide bond? And if so, how does it dimerize? And in which compartment do you think the heterodimerization occurs?
All right, so that's a really interesting question. So as mentioned, and I need to look up the literature, there is one study that I really like, and I'll try and find, if you were really interested in this, I'll try and find the citation for you. I should have put it in here, which has delved into this in greater detail. They suspect that up to, and this is where I'm like, but they suspect that up to 80% of your IgG4 might be dimerized. And it happens naturally. So people have actually done this is ex vivo the right word? In vitro, where you can actually put in different IgG4s and throw them together, and they will naturally create heterodimers. So that occurs naturally in vitro, and that's why they estimate that up to 80% of your antibodies of IgG4 in your body, because it's just naturally occurring. I'm not sure, and this is where you can reflect that I'm not a protein biochemist person, understand why they maintain and stay there together, because it does make me wonder, how is it happening in the body randomly and stochastically? That's an area that I probably need to read up more on, and I don't know the answer to. And I probably need to talk to a protein person to really delve into that.
There are also some questions online.
Oh, how do I look at them?
No, I'll just read it for you. So Felicia asked, is there anyone has assessed how stripping the IgG antibody Fc region of this N-glycan affects the structure or the function of the antibody?
So has anyone assessed how the Fc glycan?
Has anyone assessed how stripping the IgG antibody Fc region of this N-glycan affects the structure?
Oh, so stripping it completely. Sorry, that was a word I didn't get. In other words, what has been, and this is well described, is if you completely cut off and remove that glycan completely, so there are actual natural enzymes that are present in bacteria that do this, and cut it off, it actually results in the inability of the antibody to bind a lot of the different Fc receptors. So that presence of that glycan is actually essential for binding, for example, Fc gamma receptor 3. It's not for all receptors, but for Fc receptor 3 specifically, it's essential.
Any more questions? There's also a question online. Have there been any study that looked at HIV vaccine non-responders, and why do they not upregulate IgG3 antibodies?
So I assume you mean HIV non-responders. I mean people who have chronic HIV and progress more rapidly. So what has actually been observed is that in acute infection and HIV, they actually do, everyone actually, it's a very common thing, they actually upregulate high levels of IgG3 in acute infection. And then the individuals that rapidly progress rapidly lose their IgG3 over time. What is very unique is that individuals that are there's this term called elite controllers. So these are people who naturally control HIV disease. So they suppress the viremia naturally without any therapeutics. They actually have high levels of IgG3 early, and they actually maintain their IgG3 levels for longer. And have better FC effector functions. So it's not just the ability to induce them, but the ability for them not to class switch out to different subclasses. Because I didn't delve into this in great detail, but IgG3 is actually very early upstream in that, maybe it is here, in that locus. So you can see if you look at the IgG, in the human IgH heavy chain locus, you can see IgG3 is actually even before IgG1. So if you're actually class switching out into a downstream one, you can't class switch back into it. Or you can class switch down and continue downstream and downward.
I think that concludes today's postgraduate seminar. And thank you so much again for the wonderful talk.
Thank you for, invitation.